Excellent. Thank you. Um, Great. So I am going to be telling you today about some work that is a little bit different from most of the talks that I've seen over the last couple of days. I have not built tools. I am applying tools that other people have built, and I am applying tools madly, trying to figure out everything I can about a really rich and complex data set. So I want to foreground the fact that this work is work done by a very large team. So I'm showing you here a picture of about half of the team that has led most of this work. So this is the NSF-funded Emerge Biology Integration Institute, which I've had the great good fortune to be part of for the last several years. Circled in orange there are a subset of the co-lead and co-senior authors on the paper that I'm going to be telling you about. So this is really intensely collaborative work. So as we transition into the microbial session, I just want to contextualize the work that we do. So if you think about everyone here in this room and everyone we know and love and the other 7.7 billion people on Earth, think about our carbon. We can represent it as that quite small pink box. The carbon biomass in viruses is about threefold as much, in archaea about 100-fold as much, and in bacteria, well, more than 1,000-fold as much. So if you believe that human activities can shape the world, you really ought to be thinking about what microbial activities can do too. So I'm not making this up, right? We know that microbes can shape the world. They've been at it for a really long time. Microbes invented photosynthesis, and by the time land plants came along to steal it, they had already been reducing the amount of carbon dioxide in the atmosphere dramatically and increasing the amount of molecular oxygen, right? So if you enjoy multicellularity, think a microbe. So the microbes that I'm going to be talking to you about today live in the soil, and in particular in northern soils. So these soils are really interesting to us for a lot of reasons. One is that the world's northern soils store an extraordinary amount of soil carbon. Right, so you might think that this is odd insofar as the growing season is notably short in northern latitudes, but it's intense while it lasts, and at the end of it, everything freezes. Right, so this is very different from a system like the Amazon, where there's a ton of photosynthesis and primary productivity, and then everything rots. Right, but in the north, it all gets stored. It gets stored in the permafrost. And that's been serving the world pretty well for a long time. But these same latitudes are the latitudes that are projected to warm fastest under current climate models. So if we take a top-down view of the world, this is the permafrost zone of the world. Closest to the pole, we've got the continuous permafrost region. And as we move southward, we get into the discontinuous permafrost margin, and then it gets patchier and patchier and more isolated. So when warming comes to this region of the world, the effects are quite literally earth-shattering. So you may have noticed that ice is less dense than water. And if ice is what is holding up the surface of your land, then when the ice melts, the land subsides, right? And so the surface of the earth tears. This is a problem, particularly if you like to have roads or houses or any sort of infrastructure. So the site that we study is called Stordalen-Meyer. It's in the discontinuous permafrost region. And it's a really amazing site. One thing is there are roads that get you there. There's even a train, right? So this is very different from most Arctic sites where you need like a helicopter, and that does not do good things for your direct costs. But we can get there. People have been studying this site for a really long time. So there are climate records at the site going back 100 years. And if you go back even farther, there's indigenous knowledge from the local Sami people. So we've got a bunch of different sources of knowledge. It's a really beautiful place, I'm told. I haven't made it there yet myself, but it's instrumented. You know, we've got instrument huts. There's a whole field research station. It's a really very sweet setup for field work. One of the things that we have built at the site is boardwalks, which are helpful for keeping your feet dry until the land subsides, right? So we see changes in whether our boardwalks are above ground or below ground on the timescale of a single PhD thesis, right? So we see this landscape changing fast. Okay, so The climate records at the site, I'm showing you here the mean air temperature at Stordalen going back to 1913. The period that we talk about as climate normal, though we all know that this is not really normal, is in light gray. And the study I'll be telling you about today is in the period 2011 to 2017. Okay, so you can see the temperatures rising during this time. So one of the very cool things in this collaboration is that we are, as Patrick said, very interdisciplinary. I've got colleagues who do remote sensing work. And so we've got drone data that can classify the land cover at our site down to 3-centimeter resolution. So this is super, super cool. We can look and see what kind of plants there are, and then we can make predictions about what the microbes will be below the soil. So one of the reasons that it is helpful, excuse me, to be able to do this kind of plant cover classification is that as the site thaws, we can see it move through a progression that's fairly stereotypical of different habitat types. So there's an intact Pulsa type landscape, which has a very characteristic plant community. It's nice and diverse. It's got lots of forbs and graminoids. And please don't ask me more about that because I'm not a plant biologist. It is underlain by intact permafrost. And then there's what's called an active layer on top. Right, so the permafrost virtually never gets right to the surface of the land. We're studying what's going on in the active layer. And as we see thaw, that active layer deepens and deepens and deepens until we cannot poke a long stick into the ground and find permafrost anymore. So as we start to thaw from the Pulsa landscape, we move into what's called a bog. This is partially thawed, so there's some subsidence at the land surface, but it's not connected hydrologically to the sites around it, right? So it's called a perched water table. And the water table tends to move up and down with rainfall. Okay, so if you are a soil microbe, this is a super interesting and dynamic place to be, which is maybe not what you associate with bogs, but truly. As the thaw proceeds, we get into a fully thawed fen. Okay, and so here, the water table is above the surface of the soil, right? So this is pretty much constantly inundated. And again, the plant communities are changing all along this gradient. So I want to just show you a few pictures of what it looks like as we move along this thaw gradient. So in the Pulsa, if we take our quadrat and go try to tally up the plant community, we're going to find 15 or 20 different plants just in a square meter. If we pull a core sample from the soil, you can see lots and lots of tangly plant material in the core. You can see that the core is pretty dry. And if you look down the core hole, you don't see water at the bottom, right? So the Pulsa soil is dry. If we go to the bog, you can see immediately that the plant community is less diverse. There's lots of sphagnum mosses, they're really wet. The core is wet, and if you look down the core hole, there's water. If you go to the fen, you cannot really tell where you pulled the core from, right? So it's pretty easy to twist an ankle in the fen. You might wind up wearing waders and going out and like really working with the land. So in observations over about a decade, we've measured a lot of characteristics of the soil. And what you can see is that the Pulsa and the bog and the fen are different from each other along several axes that microbes really tend to care about, right? So they've got different oxygen exposure, different pH, different temperature, depending on how wet the soil is and how that buffers temperature change. So one thing that I want to zoom in on here is what's actually going on in the soil metabolically. So in general, in a soil system, in the aerobic depth horizons, you've got a bunch of different carbon processes, right? Up on the surface where there is light, you've got photosynthesis. This is going to be some mixture of plant and microbial photosynthesis. That fixed carbon is then again to get partitioned between being respired right back to the atmosphere or being fermented down in the anaerobic depth horizons. So if we move down deeper in the soil, the CO2 that's produced by fermentation might feed into methanogenesis. And there are a few different flavors of methanogenesis. There is hydrogenotrophic, there's acetoclastic, there's methylotrophic. All of these processes are strictly anaerobic. So the methane that they produce might again get consumed by anaerobic methanotrophs, or it might filter up through the soil where aerobic methanotrophs that are much more efficient have a chance at it, or it'll escape to the atmosphere. So it's really water that makes a soil be anaerobic. And so the balance between these processes is going to depend a lot on where the water table is. Okay, so if the water table is very low, like in a pulsa soil, there's just less of the anaerobic processes that produce methane that's getting emitted to the atmosphere. As we start to see thaw, the water table moves up and we see increased methane flux. So in a permafrost, in a pulsa, we kind of don't even find the water table. And so there's little or no methane flux that's coming off the intact landscape. And then as thaw proceeds, of course, we get converted into a system where there's increasing methane flux. So there are two pathways for getting methane into the atmosphere, and we worry about both of them. One is, as I've said, that you can fix carbon and then send it into a methanogenesis pathway and emit to the atmosphere. This is bad news because methane is about 28 to 30-fold more potent as a greenhouse gas than carbon dioxide, right? And so we are taking the same carbon and making it 30 times worse. Even worse than this pathway is that there's all of this ancient soil carbon that has been locked in the permafrost that is newly available for metabolism, right? So there we're not even pulling carbon out of the atmosphere before we convert it into methane. We're just injecting carbon. So one of the ways that we're able to monitor what's going on in these soils is that we have these really cool pieces of equipment called auto chambers that my colleague Patrick Krill installed a couple of decades ago. These are basically boxes. They're on hinges and they close down over the landscape for about 15 minutes every 3 hours automatically. They're piped to a mass spec in our instrument hut. And so we have this nearly continuous data, right, that's telling us all about gas fluxes from the pulsa and the bog and the fen, and also telling us about the isotopic composition of these gases, right? Because each of the different methanogenesis and methanotrophy processes leaves its own imprint on the isotopic composition. So we can take that data and we can take our land cover classification and we can do some scaling and figure out what is this system doing? Is it a net source? Is it a net sink for carbon? And so my colleague Ruth Varner led an analysis of these data streams a couple of years ago. And so looking at 1970 and 2000 and 2014 data, what we can see is that over that time, the pulsa has decreased by 11%, the bog has decreased by 11%, and the fen has doubled, right? And this is pretty bad news because that means we're going from net carbon sink to net carbon source. So we are very interested in what the microbes are doing in the soil along the way and how they're experiencing all of these changes. What's it like to be a microbe in a rapidly changing landscape? Okay, so for starters, it's like being a bug anywhere, right? You've got a genome that gives you some tools. You can decide which of those tools to express as RNA and as protein, and those decisions might change as your circumstances change. You're not making those decisions alone. You're in a population of other cells like you, and that population is embedded in a community of a diversity of microorganisms, viruses, other kinds of bacteria, eukaryotes, the whole lot. That microbial community exists within a broader ecosystem with many trophic levels. Mostly we ignore animals and fungi and protists. We kind of shouldn't do that. We're working on it. But for the purposes of this talk, we've got plants, we've got microbes, and we've got archaea. So there are lots of connections between these levels of biological organization. One of them is metabolic, right? And so there are all kinds of things that plants make and then exude through their roots to recruit and sculpt a soil microbial community. And that's a dynamic process. And microbes are making things and handing things off between each other. They're also connected by information flow, right? So there's lots of mobile elements, there are viruses, there's naked DNA, there's plasmids, there's all kinds of genes flowing this way and that way. So the original sort of dream of metagenomics, right, was that we could go out to a system, we could catalog the genes there, and we could say, haha, this is what this system does. We can scale from genes to ecosystems. And that's a nice dream, but it neglects the part where biological systems change, right? And we know that that's what they do. They've got all kinds of mechanisms for responding to changes in their ecosystem. In the short term, they can respond through physiological acclimation, right? They can make different decisions about which tools to make, what to use, what to try to do in this circumstance. In the medium term, they can respond through changes in their ecological assembly, right? Changes in who is there to contribute to the net ecosystem processes. And then in the longer term, their adaptation can change, right? They can change which tools they actually have and can choose to use. So what we're trying to do in eMERGE is close this loop to bring in ecosystems to genes scaling so that we have the whole sort of G2E2G loop closed, and we can think about how this system will respond to change and how it will change the ecosystem outputs in turn. So what is all of this going to mean for future ecosystem function, right? We have this landscape that's changing. We can see the change along axes that microbes care about. We know that they have a lot of ways of responding to change. What's that going to mean for the balance of metabolites through all of these carbon flux pathways? So we have a lot of modeling colleagues who are trying to ask this question, and they're doing a bang-up job, right? So they can take what we know about the soil microbiome. So this is a range of things from metagenome assembled genomes, or MAGs, metatranscriptomes, metaproteomes, metabolomes, all of these abiotic measurements that we make as well. They can feed these things into tools like Grodan or MicroTrait. We can infer some life history traits for these different taxa. We can try to figure out what the functional guilds are in the soil. And then we can bring in things like reactive transport modeling and abiotic conditions, put them into models like BioCrunch, and make predictions about what we think will happen in this system. We can even make it more complex by bringing in things like the canopy system, precipitation, radiation, wind, and try to predict things at the landscape scale. And this is great, right? There's really good work happening here. The tools that I'm talking about, these are like Bill Riley's work, Shem Lee, Owen Brody, a lot of really good people working on this. Unfortunately, the best models do not yet account for biological change, right? It's just too complex to bring in. And so what these models are doing is taking the current system, they're extracting parameters from it, they're using those parameters in model, and they're asking, what would the current system do now? In fact, what we want to know is, what will the system develop into? What will it do in the future? So how should our models account for biological change? If we want to ask this question, we really need to think about, what kind of biological change are we going to see? So in the rest of my time today, what I'm going to take you through is our attempt to distinguish between these two fundamentally different models of biological change in the soil microbiome as we've observed it over this 7-year period. So a couple of possibilities here. One is that you see a graduated response to change, right? As the extent of disturbance increases, you see a gradually increasing perturbation of the biological system, right? Alternatively, you could see a concerted response to change, right? The system maintains homeostasis until it hits some threshold beyond which it no longer can, and it switches over into some other mode of being. So we can talk about greater ecological resistance in a concerted change model if we push out the threshold along the disturbance axis. I just want to clarify here, ecologists will talk about resilience as being different from resistance, right? So resilience is the ability of either one of these systems to be disturbed and then return to its starting state, right? This is distinct from resistance to change to begin with. Okay, so we can think about these possible changes in outcome in a few different ways. Things that we tend to care about are questions like, who's there? What are they doing? How are they interacting? Okay, and so I want to talk a little bit about what sorts of changes we might expect to see along each of these avenues of inquiry. Okay, so if we're thinking just about who's there, under a gradual response model, you might expect to see a gradual change in community composition, right? Some of them hang out for a little bit longer before eventually giving way to a different kind of organism that's able to tolerate the new conditions. Under a concerted change model, the community looks like itself until it flips over to a different state, okay? If we think about what they're doing, we might look at the functions that the community encodes, right? And maybe what we see is that more microbes start to encode a particular set of functions that are tolerant of the new conditions, right? So there's a makeover in the toolkit at the community level. The dynamics of that makeover are going to be different depending on whether we're looking at a gradual change or a concerted change, right? And then again, we can look at how they're interacting, right? So we might think about looking at networks among the organisms in these systems to see how does the network topology change over time, right? Are they shifting alliances gradually as the disturbance gets more and more severe, or are they hanging out until they cannot? Okay, so the reason that we care about distinguishing between these two big models of biological change is that the consequences for our modeling are profoundly different, right? Under a gradual change model, our computational, our predictive models need dynamic community change with fine temporal resolution, right? We would be moving from like a pulsar state to sort of boggy pulsar to kind of pulsar-y bog to eventually bog, right? But those states would be distinct from each other along the way. By contrast, if we're looking at a concerted change model, we can use habitat type as a proxy for microbiome state, right? We can go back to our satellite data, we can skip getting very muddy, and we can make great predictions just on the basis of what the ground cover looks like, right? So this would be amazing. Okay, so here's the data that I'm going to be talking about in the rest of the talk. We have triplicate cores with 10 centimeter depth resolution dating back 2011 to 2017. That gave rise to 580 short-read metagenomes, from which we assembled about 13,000 high-quality metagenome assembled genomes, or MAGs. When we dereplicate those 13,000 MAGs at the 95% average nucleotide identity level, we got about 1,800 distinct species. That number is growing. We're working on recovering strain-level heterogeneity, but I'm not going to talk about that today. So what I'm showing you here, each of these boxes represents an OTU, colored by phylum, and I hope you can see at a glance that who's there is different in the pulsar and in the bog and in the fen. Okay, so I was talking to you before about this sort of what do we predict under these different models. One thing that we're going to do for the rest of the time is, because in our study period, soil temperature was marching upward at every depth that we studied, linearly with time, we're going to use time as a proxy for extent of disturbance, okay? And now what we're going to try to do is say, okay, well, we've got these different communities. Let's put them in ordination space and see how time progresses moving around that ordination space. Okay, so we can expect that if there is a gradual change model, we're going to kind of meander our way from a pulsar region of the ordination space over to a bog region and then eventually over to a fen region, right? Whereas if there's a concerted change model, we're going to be noodling around in pulsar space, and then we're going to have a saltatory movement. We're going to jump to a bog space, jump to a fen space. Okay, one thing I want to flag here is that that big jump might or might not get captured in any given data set, right? So we've been going out and sampling in the pulsar, we're sampling in the bog, we're sampling in the fen. We don't know whether we'll capture the movement of a patch of pulsar to bog or the patch of bog to fen. Okay, so just as a sanity check before we dove into our analysis, we went to look at what had been done in the system before. So this is community composition data from 2010, 2011, 2012, from Woodcroft and Singleton et al., 2018. And I hope you can see that there is depth structure to this data, right? So the community is different not only across the three habitat types, but also moving down through the soil within each type. And so the first thing we did when we ran our ordination is just to say, do we see that depth structure, right? So is it the case that shallow looks different from deep within each of these habitat types? And we do see that smoothly, right? So all of these points are colored by depth, and you can see a progression within habitat type from shallow to deep. If we then recolor the points by time, what we see is that there's no temporal structure to these samples, right? The pulsa is noodling around being pulsa, but it's not moving toward bog. And so this is starting to suggest that there's a concerted change rather than a gradual change happening in the system. So then we wanted to look at metabolites and to ask both, is the coding capacity for metabolic transformations changing over time? And also, is the transcriptional activity changing? So my postdoc, Derek Smith, and Sam Maroney from Ben Woodcroft's group spent a lot of quality time on KEGG looking at metabolic maps and curating what they call meta pathways, right? So not just single step transformations, but broadly, like how are we getting from here to here, right? And they worked with Rory Flynn and Caleb Orton in Kelly Wrighton's group to get those meta pathways into a custom version of Kelly's group's tool, DRAM, so that we could look at the prevalence of these pathways in our data over time. Okay, and so again, we might see a sort of gradual change in the meta pathway abundance, or we might see a concerted change. We don't need to expect necessarily in a gradual model that change would happen at the same rate for every pathway, right? We might see some pathways that change quickly and others that change slowly, but we would expect a smooth rate of transition. By contrast, if it's a concerted model, we're going to see one set of pathways until we see another set of pathways. We can also, sorry, just to flag again, that we might or might not capture the moment of transition. It gets a little bit messier when we look at metapathway transcription in metatranscriptomes. We have a smaller number of metatranscriptomes than we do metagenomes, but we do have them from 4 years. So the picture looks pretty much the same for what we would expect in a gradual transition. In a concerted transition, it's a little hard to know, right? You might imagine that an organism would need to change the extent of transcription of a given tool in order to maintain the same level of flux if you're moving into conditions under which the enzymes are getting less efficient. So we don't have as strong a prediction here for what we would expect to see. When we look at the cumulative relative abundance of these pathways in our MAGs, what we can see is that these traces are mostly pretty flat, right? We are not clear on what was going on with motility in the pulsa in 2014, weird times, but basically it's flat, right? And so what we're seeing again is that there's not a signal here for gradual change. Similarly, when we look at the metatranscriptomic data, what we can see is that there's not a strong signal through time. We can also say, okay, well, if there's not a strong signal through time, let's aggregate over time and look at this sort of holistic view of what sort of metabolism is going on in these systems. There's a lot here. There's a really big dynamic range in the data. So the data viz on these bullseye plots is kind of a work in progress. I want to just highlight a couple of different pathways. So one is nitrogen fixation, which you can see at sort of midnight to 1 o'clock. And what you can see is that the relative abundance of nitrogen fixers is increasing as you move through thaw, but they're not doing that much in the fen. They're much more active in the bog. So we're interested in nitrogen fixers in general because boreal landscapes tend to be nitrogen limited, right? And so that's going to be a constraint on the extent of primary productivity in the system. So we can also look at methanogenesis. And what we can see here is that there's not that big a change in their relative abundance. Even though we worry about them a lot in the fen, they're never going to be a major player in compositional terms, but boy, are they active, right? So there's enormously more transcriptional activity of methanogenesis enzymes as we move across the gradient. So we can also then say not just what's the pathway abundance, but also, okay, these MAGs, what kind of MAGs are they? What do they specialize in, right? Of course, we've got methanogens and fermenters. We also notice that a whole lot of our organisms have lots of possible substrates, right? So we classified as carbon generalists those bugs that can deal with at least 3 broadly distinct carbon substrates, right? And those guys dominate this scene. What you can see, I hope, is that they are particularly dominant in the pulsa, and the bog, and then less so in the fen. But you can also see, I think, that their dominance is pretty steady over time, right? So once again, we're not seeing gradual change. Okay, so then we can start to ask about interactions among these organisms and how these communities got this way. So I am cribbing here very heavily from Hannah Holland-Moritz, who's a spectacular postdoc in Jessica Ernakovich's group. Everything I know about ecological assembly modeling, I learned from Hannah. I will probably make mistakes, and they're mine, not hers. So our basic strategy here, we're pulling from Stegan et al. 2013. And this is to say, okay, well, we've got all of these different communities, and we want to do a bunch of pairwise comparisons and try to figure out whether the shift from the community at this time to the community at that time is driven more by processes of selection or of drift or dispersal or what, right? And so we can think of the selective processes as being basically deterministic. We can think of drift and dispersal as being basically stochastic. And we have this sort of stepwise process by which we can assign a relative proportion of influence for these different processes to our communities. Okay, and so we can do some beta NTI, the beta diversity nearest taxon index modeling, where we construct a null distribution for how different from each other the nearest relative in community A versus community B is. And we can ask, so in these pairwise comparisons of communities, are they phylogenetically overdispersed, excuse me, or phylogenetically underdispersed, or are they, pretty neutral in that regard? If they're neutral, we can then say taxonomically and with abundance weighting, are they more distant than you expect by chance? Are they less distant than you expect by chance? Or again, are they just neutral? Okay, and so we can apply this modeling to all of our metagenomes. And what we can see is that the ecological assembly processes shaping the communities along the thaw gradient are, number one, distinct from each other, and number two, steady through time. Okay, so the pulsa is dominated by homogeneous selection. The bog is dominated by dispersal. The fen is really dominated by dispersal. And this makes a lot of sense with what we know about the role of water in the landscape, right? So again, the pulsa is dry. It is harder to disperse through the soil. The bog is getting wet. The fen is hydrologically connected, right? So there's an advective term there that can be reseeding these communities as time progresses. So the next thing we can do is say, well, what about network modeling through time? Okay, we've got 3 cores from each site from each year. We've got 4 sections from each core. And so we can make some within year, within habitat co-occurrence networks. Okay, and then we can say who co-occurs with whom and how does that change over time. So when we make all of these networks and we stack them up, then we want to say how consistent is network structure over time. So we can look at the degree rank of each mag, each node in these networks. And we can look at Spearman's row and see how Spearman's row changes for all of these pairwise year-to-year comparisons over time. So again, you can imagine in a concerted change model that there's not going to be a major time structure to the row values. In a gradual change model, it ought to be the case that the farther apart your comparator years are, the less correlated those community structures are, right? And what you can see if we run all these pairwise comparisons is that there's not a strong time structure. So Fen 2013, again, a little weird, don't know, but basically it's steady. We can also then allow cluster membership, subnetwork membership, to vary from year to year. So microbes are permitted to enter and leave different subcommunities. And we can ask how persistent are the subcommunities that we observe in these networks. And what we see is that when we adjust for the relative abundance of the OTUs in our data set, something between 40 and 98% of the microbes in the mire belong to persistent subcommunities, right? So subcommunities that we observe in 6 to 7 years of our 7-year sample. We can also take our networks and put them together with our metabolic analysis to say, who do methanogens like to hang out with? right? When we look at methanogens network connections over the 7 years, what we see is that they are heavily, heavily dominated by generalists. This is especially true in the bog, but true to a pretty substantial extent in the fen as well, with fermenters in a pretty distant second. We can also take a totally different network approach. So we can do weighted gene correlation network analysis, where we start with a matrix of samples and OTUs, and we build networks in which OTUs are nodes, and the edges that connect them are weighted by co-occurrence. We can scale these networks until they exhibit scale-free topology. We can decompose them into subnetworks in which the OTUs have partial membership. What's useful about that is that now we have a single scalar value that's associated with each OTU, and we can plot that against environmental variables that we care about, right? And so we can say how closely correlated is OTU subnetwork membership to these environmental variables of interest. And so how good is each OTU at predicting that variable? So a variable that we care about a lot, since we're interested in methane, is what's called the fractionation factor. So I mentioned before when I was talking about the autochambers that all of the different methanogenesis and methanotrophy pathways leave their imprint on the isotopic signature in the carbon that they produce or leave behind. So we can calculate a fractionation factor that tells us about the balance between these processes, and we can go out and measure it in the soil. What we found is that the light green subnetwork, which is an arbitrarily assigned name, correlates really, really strongly with our alpha C values, and that when we try to predict alpha C values on the basis of membership in this light green module, we get the kind of correlation coefficient that ecologists simply do not see, right? This 0.78 is nuts for ecology. I wish I had a screenshot of the Zoom meeting when this plot was first up because it was drop jaws all around. Okay, so then we can look and say, well, who is in this subnetwork, right? Who is it who is so closely tied to this output of the community, right? This big sort of summative output of methane production and consumption. And what we see is that while there is one methanogen there, this signal is dominated by generalists, right? So we've got one homoacetogen, we've got one macromolecule degrader, but mostly it's generalists. And it's not just that, it's also that if we look at how tightly correlated to alpha C each of these different groups is, several of them are more tightly correlated than the methanogen. Okay, so I have gone through a bunch of different analyses. I have not gone through all of the analyses in this figure, but we have looked at the organisms, we've looked at the functions, we've looked at the organization of these communities, trying as hard as we can to find evidence for gradual change. Because that's what we expected to find, right? We thought we're perturbing the system. We can watch the landscape change before our eyes. Of course, there's going to be change underground. We looked and we looked and we looked and we did not find it, okay? So what I'm showing you here, we call this the trust us figure because as we've prepared drafts of this manuscript, every time we show it to someone in the collaboration, they say, but have you looked? Have you looked at this other level of granularity? And we say, yes, we have looked at this other level of granularity, right? So we've looked at the composition of marker genes at the phylum level and at the genus level. We've looked at the composition of the MAGs. We've looked at the marker genes at the strain level. We've looked at richness and evenness and diversity. And only in the deep fen do we see significant change with time. That's it. We've looked at the composition of metabolic pathways. And I haven't told you about this at all, but we've also done an annotation-independent look at metabolic function by looking at protein clustering, right? Where we are totally agnostic as to the content of these genes. And we're just asking which genes are there and are they changing over time? And the answer is no, except in the deep fen. And then we've looked with a couple of different methods at how the interactions in this community are changing, right? We've looked at our networks, both WGCNA and our multi-layered networks, and we've looked at community assembly, and again, no change with time here, not even in the deep fen. So we feel pretty confident at this point that we can rule out a model of gradual change in these communities. and that we can, in fact, use habitat type as a proxy for the microbiome state in this system. We're also seeing that there's greater ecological resistance than we expected, which is pretty exciting. And we think that that's driven by several different factors. One is functional redundancy, right? It's not that there's one generalist in the system, there are many. If somebody gets tired, the others can step in, right? Another thing is that they are metabolically versatile. If one of your substrates becomes less available, if one of your enzymes becomes less efficient, you've got alternatives, right? And so you can keep doing what you're doing. The other thing, particularly in the bog and the fen, is that we've got dispersal that is hitting the reset button on the composition of these communities. Okay, so the modeling problem just got easy. This is amazing news, right? We can go out with drones, we can see what the landscape composition is, we can make really strong predictions about what's going to come out of the surface of the land that summer. We can put all of this together. By the way, if you ever have the opportunity to work with Elena Hartley, hire this woman. She's a great artist and really, really helpful in helping you conceptualize, like, oh, what is the picture of our data? Strongly recommended. So we can look at how the change is happening across these transition states. We can see differences in the community. We can see differences in dispersal or selection. What we don't know is what's happening inside those transitions. And we don't know how to predict them. We don't know what the state is right before that transition. But that is increasingly important for us to understand, because it's not going to be a little, it's going to be woomph, right? We're going to go from pulsa to bog, we're going to go from bog to fen, and we're going to go from net carbon sink to net carbon source. It's going to happen fast, and we don't know how to intervene in the landscape to prevent it. Okay, so this is the work of many, many people. Some of them are in my group, most notably Derek Smith. Many of them are in the broader eMERGE and Beer Soil collaborations. And I want to just note here at the end, I was talking with Annie about this a little bit last night. These are really big teams. They're composed of people. That represents a challenge. We've been working with sociologists who are training us in how to be a good team. We've learned a lot from them, and we're now offering trainings ourselves as we go to meetings. You can't go to this one, it was last year, but we're continuing to offer them. And we have a toolkit of team science strategies that seem really to work. So by all means, talk with me about that or about science afterwards. I'm very happy to take questions.
Any questions for Sarah? Have you maybe
either yell?
Yeah, either yell or proceed to the microphone, whichever's.
I can speak up.
Great.
Thank you so much for that talk. That was amazing. I really am wondering though, so in this particular case, you showed that in this particular ecosystem with three macro states that are easily visible, the microbiome does fall into one of these three states. To what extent have you used these types of techniques to show that other microbiomes either do have this sort of gradual transition or the sort of step function?
Right, that's a great question. Absolutely none. So we've been busy convincing ourselves that this is what's going on in this system. But what we're hoping to do over the next few years is start broadening out to other permafrost sites, other permafrost peatlands, especially across the north. And then think about how do we extrapolate from that kind of system to other soil systems and then even beyond. We do think that the basic features we've identified as contributing to ecological resistance are likely to be the kinds of things you should look for, you know, in your system of interest. If you want to say, is this going to be resistant? Is it going to be changing gradually? Ask yourself, right? Are there strong dispersal terms? Are there functional redundancies? Is there metabolic versatility? And if you see those things, I think it is reasonably likely that you're going to see a system that's subject to concerted rather than gradual change. Yeah.
That was a fantastic talk. Thank you. Do you have, are there any open databases and resources you can access to if we want to play with your data?
Yes. please. We have more data than we have hands and brains to deal with. So if you go to the Emerge GitHub page, I apologize, I should have had a QR code up here, but emergebii.github.io. There's a lot of data. It is not all public yet, but by all means reach out. There are so many more questions that you could ask of this data. I haven't even put up the metabolomic data, the metaproteomic data. There's so much to do. Yeah.
In the back?
So this is from, based on my memories from a very, very long time ago. So back in the day, there was a lot about something called biomass, and then bacteria would, came out something out, and bacteria would reconfigure the metabolism so that can you, are you looking, or did you find that there's some similar principle maybe? It sounds like they would like change the condition, and there's an optimum, and at some point that optimum is not feasible because you change the conditions and they have to take something like that.
So the question, I think, please tell me if I'm wrong, is about whether we might be able to manipulate these organisms in order to manipulate the ecosystem outputs.
No, it's if you look at the metabolism and the production rate of this, that, and the other thing, for individual bacteria, people would look at something called biomass, which would be a combination of all those things that a bacteria needs, and they would try to, and you could find the state of the bacterium, so how much goes through every process, by simply trying to find the state with the maximum biomass production.
Yeah, absolutely.
There should be something similar.
Yeah, so this is most like the BioCrunch model. I'm sorry, thank you. So the question, actually correctly understood, I think, is about whether it's possible to model the effects of knockouts on biomass production for all these different taxa. Yeah, so the BioCrunch model that Owen Brody has made does a really nice job of trying to get to that sort of question, right? The expectation is that they're trying to maximize biomass. There's some new literature that suggests that might not be the right target function, but that's a bigger conversation. There is new work from Bill Riley and Jen Lee that tries to take genome-inferred traits to improve our ability to do that. And it does a pretty good job. One thing that we're really limited by, and that my lab has been trying to tackle, and work that I haven't talked about at all, is the total lack of model systems from these environments. So we don't have anything that we can poke, right? We just have observational data. And so, we've got some organisms that we're working on isolating. They tend to be slow because they like to grow in the cold, which makes it a little bit more challenging, but we'll get there.
Other questions for Sarah?
Hey, thank you for such a comprehensive talk. I was wondering how dramatic the transition of the ecosystem, like the soil itself, is. Is it so obviously fen versus bog or permafrost?
Yeah, that's a great question.
How quickly does it change?
That I don't know, right? Because when we go out, so the thing about finding out about this system by core sampling is that once you pull a core, that's it for that core, right? It's destructive sampling, and you have to go sample in a different place next time. This is especially true in the pulsa, where because it's dry, right, all the oxygen exposure, like it just percolates through. So we never know when we pull a core, how long has it been this way exactly here? I will say the change in the soil as a physical medium is very, very apparent. So we always take pictures of every core that we sample. And there have been many times over the years when we've gotten metagenomic data back and we look at the composition and we say, whoa, are we sure that this was from the bog? Because it really looks like a fen core, right? And we go back and we look at the picture of the core and we can see, was this really a bog core or was this a fen core? And that's everything from how wet the soil is to the color of the soil to how dense the plant material is. We have a lot of familiarity with the physical structure of the soil because in work that I haven't talked about here at all, we've been running microcosm incubations in my lab, doing temperature perturbations of material taken from the fen and from the bog. And I have to tell you that when you tweezer a kilogram of soil into narrow mouth serum bottles, you have a very fine appreciation for the physical structure of the soil. So yeah, we can see those differences, but we don't know how quick the turnover is.
Thank you.
Great. Any other questions? I have one actually. So I was wondering, you talk about these generalists, right, that are kind of like in this correlated group with the methanogen, right? I was wondering, do we have any intuition about like what aspects of their metabolism are important? Like, or is it like more that there's just so many different ways to get like a source of, you know, something that the methanogen can use, whether that's hydrogen or something else?
Yeah, so I think part of it is just production of small organic acids, right? So if I go back, hang on. Yeah, okay. So I didn't walk through all of the functions that are documented here. All of these guys are CASEs, right? Carbohydrate active enzymes. The soil in the permafrost, in peatlands, is really weird. Like you might think of soil as a basically granular material, but no, right? This is dead plants. So there's a whole lot of really tough carbohydrate material, and the ability to break that down into the small organic acids that are substrates for methanogenesis is really critical.
Yeah.
Just,
Yeah.
could you go back to your square trees that represent the composition?
Yes, can you tell me when I get there?
Sure, it's the one that shows which microorganisms, which phyla are present.
Yes.
It was pretty early.
Yep, sorry.
No, maybe I should see, I'm sorry.
No, you're fine. Nearly there. Yes?
No.
Oh, the tree maps. Okay.
The tree maps.
Yes, I have no idea why they're called tree maps, but yes, these guys?
Yes.
Great.
So do you have any, I mean, your generalists are sort of spread across all of these taxa?
They are, and I should have put in the Secret Ninja slide that shows a tree map by specialization. I can pull it up and show it to you afterwards, but I don't have it in this slideshow.
And this is only bacteria, not archaea?
No, this includes archaea.
Okay, yeah.
Yeah, by the numbers, it is much, much more bacteria than archaea, but they come up. So all of the methanogens are archaeal. They're helpfully named Methanobacteriota, but they're archaea. So they are the purple guys in between the sort of gray and green. And then in addition, the disulfos are actually bacteria.
Yeah.
But there's another archaeal phylum here that I'm totally failing to identify.
Thank you.
Sure.